విద్యా విధానంలో డాక్టర్ బిఆర్ అంబేద్కర్ ఓపెన్ యూనివర్సిటీ దేశంలోనే మొట్టమొదటి సార్వత్రిక విశ్వవిద్యాలయం. దాదాపు 2.5 దశాబ్దాలుగా దూర విద్యా విధానం ద్వారా కంప్యూటర్ కోర్సును అనుసంధానం చేస్తూ BA, BCom, BSc డిగ్రీ కోర్సులను అందిస్తోంది. పోస్ట్ గ్రాడ్యుయేషన్ స్థాయి, డిగ్రీ, డిప్లొమా స్థాయి కోర్సులను ఔత్సాహికులైన విద్యార్థులకు అందిస్తోంది. డెవలప్మెంట్ స్టడీస్లో సెంటర్ ఫర్ ఎకనామిక్స్ అండ్ సోషల్ స్టడీస్ ద్వారా MPhil, PhDతో పాటు విశ్వవిద్యాలయమే స్వయంగా దూర విద్యా విధాన అంశంపైన PhD వంటి పరిశోధన కోర్సులు ప్రవేశపెట్టింది. సుమారు 4.5 లక్షల మంది విద్యార్థులు ఈ విశ్వవిద్యాలయం నుంచి ఉన్నత విద్యావకాశాలు పొందుతున్నారు. డాక్టర్ బిఆర్ అంబేద్కర్ సార్వత్రిక విశ్వవిద్యాలయం స్థానికంగా స్వయం బోధనా పద్ధతిలో ముద్రణ రూపంలో రూపొందించిన కోర్స్ మెటీరియల్ అందిస్తోంది. కోర్స్ మెటీరియల్ అధ్యయనానికి సహకరించే విధంగా రేడియో, టెలివిజన్ పాఠ్యాంశాల ఆధారంగా కార్యక్రమాలు ప్రసారం చేస్తోంది. అధ్యయన కేంద్రాల ద్వారా ఏర్పాటు చేసే సలహా సంసర్గ తరగతులు, సైన్స్ ప్రాక్టికల్ సౌకర్యాలు విద్యార్థులకు స్వయం అధ్యయనానికి ఒక చక్కటి అవకాశం. ఇవి విద్యార్థి సమీపంలో ఉండే అధ్యయన కేంద్రం నుంచే పొందొచ్చు.
Good afternoon to all the viewers. The topic for today's discussion is herbarium methodology, and we have with us subject experts Dr.B.Rajani, Assistant Professor, Department of Botany, PG Science College, Saifabad, Osmania University, and we also have another expert, Dr. Usha Kiran, a Senior Faculty of Botany Department, Sri Gayatri Educational Institutions, Kukatpalli. We welcome the subject experts and we will have a discussion on herbarium methodology. First, we shall discuss what is a herbarium. Dr. Rajani, can you elaborate on what is a herbarium?
Yes. Herbarium is a storehouse of plant specimens collected, dried, poisoned and mounted on appropriate sheets called herbarium sheets, and then arranged in sequence according to known system of classification. The word herbarium was originally coined to a book which is about dried medicinal plants. Herbaria in Latin referred to slender, weak stem plants. A collection of herbs was known as herbarium. Tonfort in 17th century used the term herbarium for dried plants, and this method was previously known as hortus siccus.
Dr. Usha, can you give us the origin and history of a herbarium?
The Leuca Guinea in the year of 1490 and 1556 is the duration of a person who was a professor of University of the Bologna. Italy was the first person to dry plants under pressure and mount them on the paper, and he recommended this method of preservation. He paved the way for the establishment of the first herbarium of the world in Italy. The oldest preserved herbarium specimen was prepared by Gerardo Cibo, who was a student of Leuca Guinea. And origin and history point of view, in going in a detail about of this, in the beginning the plant specimens were mounted on the sheet of the paper which were bounded into books and the volumes, and Linnaeus arranged these herbarium sheets according to various groups following the taxa groups, that is a species or the genus level organization in the form of a groups. The size of the sheet was according to the size of the plant. The present concept of the herbarium has been changed in a mode of collection, pressing process, drying and preserving the plant specimens. And herbarium, especially in case of the Europe, the Linnaeus had been elaborated how to prepare it first time.
Then next we have to see why we should have a herbarium and what are its functions. Dr. Rajani will tell us.
Yes, herbarium is a conservatory of plant material and storehouse of data. The simple form of indexing the plant specimens in the herbaria helps to retrieve the data very quickly. It is the basic tool for taxonomic studies and serves as fundamental source in identification of plants. It is source for the search of new genetic material and used for extraction of DNA also. It serves as a teaching material in the classroom. In pharmaceutical research, it serves as resource for identification of crude drugs. Herbarium serves as a source for biodiversity information also. It helps in phytogeographic studies also. Databases of plants are developed with help of herbaria, which is called electronic herbaria.
Now that we know what a herbarium is and what its functions are, I think we should study what are the different kinds of herbaria.
There are two types of the herbarium, that is a general and as well as a special type of the herbaria. And the general herbaria had been categorized into three, that is international herbarium, national herbarium and the local herbarium.
Now these herbaria, they require the plant material, and usually from which parts of the globe can the material come from?
Usually these plant material, the specimens can be brought from North and South America and parts of Africa and even from Australia.
Now these herbaria, we have seen that they are international and national herbaria. So Dr. Usha will tell us what are international herbaria.
Yes, ma'am. It is international herbaria is a very large herbaria. Nearly 4 million specimens were collected from different countries. They are rich in type specimens. They help in writing floral monographs, in identifying rare specimens, new taxa, that is providing facilities to the visiting botanist. First herbarium in the world in the year of 1570 in Bologna, Italy by Luca Guini had been introduced this thing. And international herbaria, presently around 4,000 herbaria, over 165 countries had been preserved and conserved. In this, index herbarium is a world catalogue of public herbaria published by the New York Botanical Garden. And as well as this international herbaria are providing a loan specimens to other reputed herbaria on request. And here in this particular institution, the Paris of the Herbarium, this is the first world's largest herbarium, Natural History Museum of Paris, established in the year 1635. 9 million specimens were preserved and conserved for the future reference. Royal Botanical Garden, Kew at London, is the second largest herbaria in the world, and Royal Botanical Garden had been located Kew at London, established in the year 1853. 7 million specimens had been conserved for the future reference point of view, and some are type specimens we are calling. These type specimens are nothing but like species level as well as the genus level collection had been completed, that is around 3,50,000 type specimens. Third, at the 7 million above, pressed plants are kept at Kew at London, and this is one of the largest herbarium. And third herbarium is New York Botanical Garden. New York Botanical Garden, established in the year 1891, 6 million specimens were conserved for the future reference.
Now we shall learn something about our national herbaria.
Yeah, national herbaria consists of the plants of the country, also covers the neighboring country specimens. National herbaria are old and contain good number of collections. These play a significant role in writing national and regional floras. Provide facilities to visiting botanists also. Major herbaria in India are managed by the Botanical Survey of India. The first largest herbarium, the Central National Herbaria, Howrah, established in 1793 with 2 million specimens. The next one is Forest Research Institute, Dehradun, which is established in 1816, has 3,30,000 specimens. The Botanical Survey of India, Shillong, established in 1956, has 2,60,000 specimens. The National Botanical Research Institute, Lucknow, established in 1948, has 1,20,000 specimens. The local herbaria deals with region in a country such as state, district or nature reserves. They have short history and contain few types of specimens. These are useful in production of local floras, for example district floras. They cater to the needs of local educational and research institutions. Southern Circle Herbarium of Botanical Survey of India, Coimbatore, established in 1874, has 2,59,000 specimens.
Then we have some special herbaria, and Dr. Rajani will enlighten us on the special herbaria.
Yeah, these special herbaria has very limited scope, like historic herbaria, which are very rich in type specimens. For example, algal herbarium at National Institute of Oceanography, they deal with algae, and even it is there in Goa also. The forest herbarium at Forest Research Institute, Dehradun, also deals with the special herbaria. Herbaria of limited scope means these are associated with large herbaria or institutions or universities. The herbaria for teaching purpose also cater the needs of teaching and research, contain surrounding area plants and economically important plants also. Sri Krishna Devaraya University Herbarium at Anantapur is one of the special herbaria.
Now that we have learnt what a herbarium is, what is its origin, its history and the different kinds of herbaria that we have, we need to learn how to make a herbarium. Now this making of a herbarium involves many steps. We will start with the first step. Dr. Usha will enlighten us on the collection of the material for the herbarium.
There are 3 steps of collection of the material. Number 1 is collection trip. The collection trip should be in a short duration, 1 to 2 days. Nearest areas should be selected. Second step is exploration. For floristic study in the different seasons we have to perform, that is taking a lapse of 2 to 4 weeks duration. And third step is in the collection of the material is expedition. Expedition is a remote or the difficult areas where we are choosing that will be takes almost several months. And precautions while collecting a material should be followed, like moving to the field work should be careful. Planning and arrangement is very important, have to be made for stay, and the cooperation from the local authorities also. And identity card should be weared by the collector, and route map should be made, and earlier published flora should be referred, and field trips in the morning slots we have to complete, and it should be completed by the afternoon slot. We should not extend towards the evening slots. And in this particular picture you are finding a collection process in the remote areas after doing in the afternoon slot. They will be sit together and discussing about that particular collected plant material and all, writing on it particular notes. That is very much essential nearby the field area.
Now in a field we have to collect the plant specimens, and what are the requirements?
The materials for field work. So whenever we are going to a field work, we need to carry the following ones: the plant press, the field notebook, the collection containers, vasculum, polythene bags. And there are some implements also, the tools which we need to take along with us to the field. These are cutter, pruning shears, scissors, knife and a digging tool, measuring tape, pencil, pen, scale, hand lens, camera, blotting paper, plant presser, ropes, identity card, compass, batteries, binoculars, altimeter to measure the altitude, preservatives like formalin, and even the first aid box, which is most important.
While collecting the plant material, we should take into consideration certain aspects which are very important. Usha ma'am, can you enlighten?
Yeah. Collection of the specimens, when we are doing, the material should be in a good condition. Herbarium sheet size must be like 28 into 42 centimeters. And the plants had been categorized as per the Aristotle onwards and the Theophrastus onwards, like herbs as well as wooden trees as well as. Like we are finding shrubs, herbs as well as the trees. In a different manner we are collecting the plant material in a different way. Like herbs, for example, here the whole plant with the root or the underground parts should be collected. And woody plants, in case like we are using a pruning scissors for cutting the leaves from the woody plants, and reproductive parts such as flowers and the fruit should be collected, and grasses and the sedges, if we are collecting, whole plant should be collected. And collection should be contain flowers and the fruits. Better to collect 4 duplicates for the single field and from other areas also. Serial number should be made, and the same number should be made on the duplicates. For example, 10 number of the plants had been collected. Its duplicates will be 14 number, should be comprise a similar number for right from the first plant to the 10 plants, should be specified in a sequential manner. And as well as we are finding various habitats like monoecious plants are there and the dioecious plants are there. We have to concentrate on the sexuality of the plants also. Unisexual flowers and the bisexual flowers should be collected in a sequential manner, and it is very important precaution while collection, because as per the Bentham and Hooker classification, we have to concentrate not only on the morphological characters as well as the reproductive characters are also very important for the accurate identification. And again, after collection of the material, serial numbers what we had made and recording the notes in the field notebook, the specimens are kept in a vasculum or the polythene bags or in the field press. Vasculum is a one tin like aluminium foil tin which had been painted with the white paint, and it should be kept in a field condition, because it may not be left off due to the white color will be deflect the like radiations or the sunlight and all. And this aluminium foil is very heavier to carry from one area to another area. That's what nowadays we are using polythene bags or a field press which had been prepared with a mesh.
Now there are some special plant parts which need to be collected very carefully. It can be the ferns or the succulent plants, or it can be the seeds, or it can be the slender aquatic plants. Now how are these plant parts collected and preserved?
Yeah, it's an interesting question, Angela. Collection of special plant parts are done like for succulents, because the succulents have fleshy tissues. They need artificial heating and hollowing of the tissue also before they are gone for the processing. The ferns, which belong to the Pteridophyta division, they have rhizome with scales and hairs which are of taxonomic value. So these are to be collected, and then fronds with sori are to be collected, and more important is the exact size is noted. And lichens and bryophytes are very delicate because they do not have mechanical tissue. These show very delicate parts, so these are to be collected in a small envelopes, and which are of stiff paper. The slender aquatic plants are to be collected in sieve or in a muslin cloth. Plants with mucilage, gums, resins, as they are very sticky, they are to be collected in folded muslin cloth. Very large plants like palms and etc., as we cannot mount them on paper, they are to be photographed, and the sizes and other things are to be recorded. The seeds are collected in an envelope, especially they are for the identification purpose.
Then while collecting the material, we need to make a note of when the material is collected, how it is collected, what are the environmental conditions under which it is collected. For that we need to maintain a field notes. Dr. Usha, can you tell us something about the field note and its maintenance?
Yeah. Field notes, after the collection of the material, detailed notes should be entered in this field notebook at the time of collection in the field itself only. The field notebook has hundreds of leaves and the pages are serially numbered, called as field numbers. Each page of the field notebook has 4 to 8 detachable slips. These detachable slips should be numbered with the tags and it should be ties to the specimen for the accurate identification. In this picture we are finding a field notes, and the field note pro forma should be possess date of collection, the date where we are visiting a particular field area and all, and the genus name or the species name or the family name. If we are well known in that field area, we have to notice. If it is an unknown plant, after coming inside the laboratory or the particular headquarters, we have to notify the genus and the species and the family name. And vernacular names and uses, it should be verified by the local persons. For example, the mandara, if collected in the field area, it had been called as a dasani in the different areas, like, and some people will be called it as an hibiscus, and some people are calling as a China rose, or otherwise called as a dasani. Different vernacular names are available. For the proper identification point of view, vernacular name should be listened properly and their users are also notified from the local persons, because local persons know very well about their users. And the locality of the collection is very important to find out the particular situation of the climatic conditions and all. Habitat is also very important, and type of inflorescence, what type it is, either racemose inflorescence or the cymose inflorescence or simple raceme it is. So we have to identify special type of inflorescence and all. And color of the leaves are also variegated leaves are there, and some are normal natural green in color, and flowers and the fruits are also vary in their colors and all. So and their shapes are also vary, as the alcohol treatment is very important then and there to decolorize the leaves for the preservation. And presence of aroma is also, for example, in the labiati member, aroma is very important in the collection of the plants, tulasi or the other plants what we are collecting in the area. In the tulasi itself only Ocimum sanctum is there, Ocimum basilicum is there, different species identification due to the aroma only we are identifying. And latex is also very important to understand in a particular aspect. For example, Asclepiadaceae members having a latex product. So in case in that particular family, latex had been identified, and abundance and frequency of that particular specimen should be identified. This abundance and the frequency is depending upon the quadrant method. So this is a Ronquier frequency method and the Ronquier abundance method in ecology, what we are studying in that aspect. In the PG level, we are finding this Ronquier classification also. In that abundance and the frequency is very important to notify this quadrant method. And associated species should be reported for the comparison point of view. In a particular local area, one plant if existing, similar conditions maybe we are finding in other local area, and this comparison will be gives accurate identification for the particular collector who are doing the work in the field area. And uses of the specimen, what it may be we collected in the field area. So and then specimens collector's name should be written in the full form. It should not be shortcut. It should be written in the full form.
Now that the plant material is collected and a field notes is maintained, now the material which has been collected, it needs to be processed in order to preserve it for a very long time. Dr. Rajani will tell us the methods of processing the collected specimens.
Yes. The processing of specimens deals with poisoning, drying and pressing. The poisoning of the material is done by two methods, that is dry method, wet method, depending upon the chemical used. The drying method is once again of two types, that is natural and artificial. Here in the photograph, the collected plants in the field, you need to press and dry them straight away to keep them in good condition. That's what is shown.
Then we have to see how these specimens are preserved. Dr. Usha will tell us.
Yeah, the specimens had been preserved, as Rajani madam had already explained about the methods what we are preserving and all. Like poisoning process is one of the method in that to avoid the deterioration and the damage. While we are collecting in a wet condition, surely some of the deteriorations or the bacterial or the fungal effects maybe takes place in the field area. So duly poisoned for the long duration of the time. And poisoning will be kills the plant immediately and prevent the formation of the abscission layer, and leaves and the flowers will not detach from the plant due to the poisoning process. In this we are finding two methods, that is dry method and the wet method. Dry method had been done by dipping the whole plant in a saturated solution of mercuric chloride in a ethyl alcohol. 20 grams of the mercuric chloride should be dissolved in 1 liter of alcohol. Mercuric chloride is a corrosive for metals. Hence, enamel trays are utilized in the laboratory. Deadly poisonous, hence disposable gloves must be used. And lauryl pentachlorophenate, that is 3.75%, should be mixed with the white spirit, safer than the mercuric chloride, and this lauryl pentachlorophenate had been sprayed on the surface of already pressed material also. That is a very good utility. Whereas in the mercuric chloride method, we may find some of the dangerous condition consequences due to the utilization of mercuric chloride. And lauryl pentachlorophenate is a very safer for the laboratory persons who are working on the specimens and all. And in this picture you are finding a picture. In this picture and poisoning the specimen, wet method. This method is suitable for the tropical countries. Day collections are numerous certain times. Not always possible to dry the collections by changing of blotter papers or the newspapers continuously. The collections are spread out in old newspapers. This is very good utility of a field worker who are doing the work, because the old newspapers are available to along with anybody. So therefore old newspapers and bundling it up at then and there only, and each bundle is placed in a large polythene bags, and 10% of the formalin is poured over the bundle. So the bundles get soaked thoroughly. The bags are then airtight, and the formalin should not be in a more concentration and the more in amount than 10%. No change of folder is necessary till reaching the headquarters. And this poisoning method, under it one more method is wet method. By this method collections can be made even over 3 to 4 months. So on reaching the headquarters, the bundles are opened out and the specimens are exposed to the atmosphere to drive away the excess formalin what we had utilized, and it will be comes out like a fume. And specimens are spread out for pressing and drying.
Dr. Rajani, can you tell us how the material is pressed after the treatment is carried out?
Yes. Pressing of specimens means the process of placing specimens between the absorbent of pressure under moderate pressure. Main object of pressing is to flatten the dry specimen, flatten and then dry the specimens. Pressing is done by keeping the straps very tight and by changing the blotters every day for 6 to 10 days depending on the weather. The plants lose their moisture and finally become completely dry. Herbaceous specimens should be washed with water to remove mud from the roots and the main plant. The main reason to show the flower how to press it, the show we have to show the front and the back of the flowers if possible. We have to open the mature flowers out so that the inside is clearly visible. Then if there is plenty of material, some of the material is kept in an envelope.
Once the material is treated and then the pressing is done, it needs to be placed on a herbarium sheet, that is mounting we have to do. Now how is the mounting of the specimens carried out? Dr. Usha is going to enlighten on this.
And this is one of the herbarium sheet, how the naturally in several colleges and the universities we are using, and where fixing the already processed material what we are finding over here, like this is a material Tephrucia purpurea fabaceae member, already had been collected with the flowers as well as fruits and vegetative characters as well as the floral characters are as we are finding. And this process, fixing the processed plant specimen on the herbarium sheet, is called as mounting. A standard herbarium sheet is 28 centimeters into 42 centimeters and usually made up of heavy, long lasting white handmade paper or card sheet.
Now, can you tell us what precautions we have to take while mounting the specimens?
Only one of the specimens should be placed on a sheet. Like already I had shown one of the sheet now lively, and the lower part of the plant should be at the base of the sheet. Space should be left on right hand bottom for the herbarium label. So the number tag with the plant is pasted in a proper position along with the thread. While mounting, the specimen should be neatly uniformly spread and fixed on the sheet, and all the parts of the plant should be easily visible for study. The specimen may be secured with the line and thread using needle at places. The label usually of 8 into 12 centimeters at the right hand side with the details like scientific name of that plant, family name, location, habitat, collector's name, collection and numbers, date of collection, local name and local use.
The next step is sorting of the specimens and filling. Dr. Rajani will tell us.
The arrangement and maintenance of herbarium deals with sorting and filling of specimens. Before the mountain specimens are incorporated in the herbarium, they are stamped with seal of the institution and an accession number is given. Stamping is done on the top right hand corner with a rubber stamp. This stamp carries the name of the institution and space for accession number and date of the accession. Sheets are listed in accession register along with their number and then incorporated in the main herbarium. The mounted, identified and accessioned herbarium sheets are incorporated in almirahs in pigeonholes which are sorted out family, genus and species wise. Same species sheets are placed in lighter covers and named as species cover or folders. All the species belonging to the same genus are placed in a heavy paper folder called genus cover or folders. Species cover and genus cover of folders useful in protection of specimens for easy reference and for convenience in handling. The sorting and filling specimens still continues, wherein arrangement of families and genera of angiosperms in according to Bentham and Hooker classification. Other groups of plants have to be arranged according to standard system of classification. All the duplicates of the specimens are maintained as stock, useful to study palynology and anatomical studies. Duplicates are maintained separately, not in the main herbarium room. This picture shows the species cover or the genus cover folders. Here the number assigned to the species is shown and the number assigned as jacket in this species.
Then next, the indexing of the specimens has to be done. Dr. Usha, can you tell us about the indexing of the specimens?
Yes. Indexing of the specimen is should be done with reference to identify the species of interest. Easy reference is very important to identify the species of interest. It provides abstract of the specimens preserved in the herbarium. Index card containing information of the specimens, field number, date of collection, localities of collection, name of the collectors, field number in the sense, for example, if you are choosing one of the state or otherwise one of the district, a particular field what we are selecting is very important to note, in that we are making like one rectangular quadrates and making a number of the plants what are existing in that quadrate area. And the date of collection is also very important, because after preservation what we are finding certain conditions are required for the date collection, duration of the time of the preservation and all, how much time is needed for the drying in a natural method and the artificial method. And the duration of the time will be vary in case of natural method, like 15 days as mentioned by the Dr.Rajani madam. And date of collection, regarding it, the drying process may be taking a lots of time. So regarding it only till date, from the starting date to the ending date, what is the consequences of the changes in the vegetative parts what we had collected and the floral parts we had collected in the locality. And localities of collection may be temperate regions are there, subtemperate regions are there. Throughout the world we are finding a climatic fluctuations. So the locality choosing is also very important, and from district to district the flora is quietly changing as per the environmental conditions. Name of the collector is very important, because for the future reference point of view, a plant in what condition they had collected. Nowadays we are using computerized databases or being maintained in the herbaria. So these computerized databases are linked with the www. Like it is a world wide web we are utilizing. No need of going every time to the field work. Certain times for the future reference point of view, within the computer system itself only, we are finding some of the information regarding the relevant particular plant what we had collected in that field area. And later on, indexing the plant proforma cards we are utilizing, and this card is comprising a locality including the country or the district or the area what we had chosen, and habitat of that particular plant, and altitude. Field identification is very important. Collector's name, that is for example, so far we had seen several classification like natural system of the classification and the artificial system of the classification. In a taxonomy we are studying that, and this collector's name, why it is very important. Any plant name what we are identifying, genus or the species epithet. First time the ICBN had proposed this particular rules and the regulations for the identification of the plants. This herbarium is also one of the key for the identification of plant. Not only the botanical gardens what we had explained so far, and even the herbarium sheet or the key or the couplets leads what we are using is each and every authentic statements are very essential, followed by the genus, species, and then scientist name. For example, Mangifera indica Linn we are using. Linn is in an abbreviated form. So collector's name, if we are mentioning in a full form, after analyzing the particular plant identification and all, genus name should be start with the capital letter and species name should be start with the small letter, followed by a scientist name should be abbreviated, and then we are utilizing abbreviation and then punctuations. So this regarding the ICBN rules follow up point of view only, we are collecting the name of that person, because plant identification is not a very easy process, because the ICBN should be accept this particular identification process what we had taken up on the herbarium sheet. Collector's name is mentioned in an accurate manner. This is a conclusion regarding the collector's name in an index card. And date of collection. Date of collection already I had explained to you. This date of collection should be collected time to till ending time. So because in that duration itself only we are processing the material as well as pressing the material, and this date of collection regarding this steps what we had mentioned previously, that is collection, processing, and then pressing. So this pressing, drying, wet method and the dry method what we had analyzed so far, this is requiring lots of duration. So date of collection regarding it is very important to notify in the index card. Plant description is also very important. Plant description such as morphological character, vegetative characters and the floral characters, very important for the classification.
Certain plant specimens, they need special type of arrangement. Can you elaborate us on the special arrangements made for certain specimens?
Yeah, they are called as type collections. They are very valuable, not referred during the routine work. They are kept in fireproof steel almirahs. Each type sheet wrapped in a folder marked with red label, red margin or red band. As you see in the picture, you can see the type specimens is preserved in this way. The special arrangement for certain species continues. The segregation of cultivated plants, wherein specimens of cultivated plants of a region should be separated from other and are kept in genus covers of a different color. The segregation of seeds for students and the beginners are completely different. Once again, we have to maintain a set of collection of species commonly encountered in surrounding area separately. There are undetermined specimens. If some plants are not identified, and these should not be mixed with the others, the regular ones, but should be kept in separate bundles for further study by the specialists. There are bulky herbarium, large specimens such as bamboos or large rhizomes and fruits which cannot be mounted on mounting shoots and require special drawers or cupboards. Then handling or sorting of unmounted duplicates should be preserved as stock. Once again, bundles of duplicates should be fumigated and stored in a closed or open racks. Duplicates should be packed in old newspapers and bundles arranged according to serial numbers. Duplicate bundles should bear the labels or tags.
Once the herbarium sheets are prepared, that is, the mounting of the specimen is done on the herbarium sheet, it has to be preserved for a very long period. Therefore, decontamination of the herbarium sheets along with the specimen has to be done. Now, how can the decontamination of the herbarium sheets be done?
Yes, ma'am. Decontamination method is very important to preserve the plants in an accurate manner, because worldwide herbaria or the national herbaria or the local herbaria or the special herbaria what we had discussed right now should be in a decontamination condition itself only, because if they are contaminated, again we cannot utilize those plant specimens for the visitors also, and as well as some research scholars who are doing the work on taxonomy for the identification of plants in the various districts to study the flora. So decontamination method is very helpful. This can be done by heating method, fumigation method or a deep freezing method, or by using the insect repellent. Specimens are attacked by the pests like silverfish and the beetles. Pest control method can be done by heating the sheets at 60 degrees Celsius for 4 to 8 hours in a heating cabinet, or kept in a deep freezers at 20 degrees Celsius to 60 degrees Celsius. Microwave ovens can also used for pest control, and pest repellent chemicals are also we are using for controlling the for controlling the fungus or the bacteria or the insects. Pest repellents we are using, that is para-dichlorobenzene and naphthalene. Both should not be used at a time because both are highly toxic. That's what para-dichlorobenzene and naphthalene should be utilized one after the other, and both should not be used at a time. Toxic chemicals, care should be taken with the time of exposure. more duration of the time we should not utilize these chemicals for decontamination of any specimen. And decontamination method, one more method is fumigation. Fumigation is nothing but volatile poisonous liquids we are using, and methyl bromide, carbon disulfide or carbon tetrachloride proportion should be 3:1 ratio of ethylene dichloride and carbon tetrachloride is used in the herbaria, placed in a small saucers or petri dishes in the herbarium case, and cases are kept close for a week.
The herbarium preparation technique is a very tedious process, and it is a very difficult task, and the herbarium users, they should follow certain instructions while using a herbarium. Dr. Rajani will tell us the general instructions while using a herbarium.
Yes, the general instructions for herbarium users: do not shuffle the sheets kept in folders. Specimens are brittle and they easily get damaged. They have to be stored, like we need to store the specimens properly, and do not crowd too many specimens into a single box. Do not write anything on the herbarium sheets, and do not lay heavy objects on the specimens lest they break. No flower or fruit is to be taken from the dissection from the herbarium sheet. Specimens which need repair should be noticed immediately. While handling full bundles, should be taken out from the racks, and pulling one or more sheets may damage the specimens. Specimens should be kept on table, should not be kept on table for longer period. They should be removed immediately after the use. They may be exposed to infection by pests if they are put for longer hours on the tables. Almirahs should be closed after taking the herbaria sheets and while keeping back the bundles also. Open flames and smoking is strictly prohibited in the herbarium room or the near the almirah. Incorrect identification should get noticed. Only an expert will make the corrections.
Dr. Usha, can you tell us the precautions once again while collecting the plant specimens?
And here the precautions point of view, from the field work to the laboratory work what we are making, this is a very important to note that precautions are should be taken. Do not shuffle the sheets kept in a folders, and specimens are brittle and easily get damaged. Brittle in the case woody plants, for example, if we are collecting, it will be dried off and may be broken certain times if we are not collecting proper way. Do not shuffle the sheets, I said, like sheets kept in a folder from one side to another side. And when we are arranging the sheets, it should be in a systematic way as per the Bentham and the Hooker classification or the phylogenetic system of the classification, that is a Taktajan Shipno 2002 classification. As per that we have to follow while we are organizing all the specimen sheets. And store specimens properly. Do not crowd too many specimens into a box, because the pressing process, for example, combs, Aroidaceae members, especially Aroidaceae, best example is Cocrse. Cocrse, if we are collecting complete plant, we are collecting. So this plant, if we are not collecting in a proper way, like complete plant, if we are not collecting vegetatively, it have the capacity to propagate. That's what we have to collect the plants in a systematic way, store specimens properly, and do not crowd too many specimens one on another. It will be damage entire specimens which are at the downstairs. Do not write anything on the herbarium sheet while you are pressing, and this pressing process is should be done with the glue. This glue is obtained from the Arabica gum we are calling. This will be available in the Telangana as well as Andhra Pradesh markets, and otherwise if you are not getting this, Fevicol also we can utilize, but should not be utilize any tape. Any tape we should not utilize while pressing on the surface of the herbarium sheet. Do not write anything on it, just press the plant and preserve for the certain period of time. Do not lay heavy objects on specimens. Of course, for the proper arrangement of leaves as well as fruits, flowers, everything should be in a correct way, because this particular plant specimens what we are collecting not only useful for the sake of what normal paleontology or the anatomy point of view, even recent biotechnology as well as cell biology point of view. If we are not finding a excessive number of the flora, like some specimens or the species are extincting nowadays, biodiversity goes on extincting. So this extinction process is revealing that we have to save some of the plant specimens for the future reference point of view. DNA extraction may be useful for what? Propagating the number of the plants in a biotechnology method by using the PCR and all. So we can increase the extincted specimens by preserving them in a proper way. So therefore heavy objects or something else if you are keeping, again and again we have to go for the field work, and it is a risky job again. Some are what short trips and some are long trips. As I said, expedition is a very lengthy process, such as an Alamalai forest, for example, if you are taking as an one of the field area. So this forest area is also very critical situation. Field area is also very difficulty conditions may be leading. That's what collection process, after doing it, we have to preserve in accurate manner. We should not misuse the materials when we are bringing towards the headquarters or the laboratory. No flower and the fruit is to be taken for the dissection from the herbarium. And this flowers and the fruits, if you are taking for the dissection point of view from the specimen copy, again for the reference point of view, what is the genus and what is the species we cannot hypothesize. And these specimens which need repair should be noticed. Otherwise any part like flower, if detached, we cannot identify the plant genus or the species, because the species characters are depending upon the characteristic features of flower as per the linnaeus, stamen number and the gynoecium, within that carpels number and the number of locules, syncarpus condition or the apocarpus condition, fused condition of the carpels or the free condition of the carpels, number of the petals, number of the sepals, sepals fusion as well as the petals fusion and all. Petals and the sepals may be free, polypetalous, gamopetalous. These conditions, technical terms, these all are. So these conditions should be what identified, and it must be available on the specimen copy. Anyhow we are maintaining duplicate copies, but even though what the original copy is there, it should be conserved for the long tenure of the time. Specimens which is what we are using, handling full bundle is very important, should be taken out from the racks, and pulling one or more sheets may damage the specimens. Already Rajani madam had told, like specimens should be maintained in a type specimen manner, like almaras we are maintaining. Like in Anantapur, one almara, how they had managed in the Open University books, we are finding this almara diagram also. Like this almara is maintained in the Anantapur floristic maintenance and all. This almara should be possessed some racks and all, preserved in a proper way, and from which type of the species, which type of the genus had been notified, and this type specimen, type genus what we are maintaining, it is very easy to take out the type of the species, type of the genus. And specimen should not be kept on a table for the long period of the time. Otherwise again what the decontamination precautions I had mentioned to you, it will be utterly flopped. And almara should be closed after taking and while keeping back the bundle. It is very important. And open flames and the smoking is strictly prohibited in the herbarium.
we have studied the various steps involved in making a herbarium. They start from going to the field, collecting the material from the field, processing the material, drying the material, treating the material, indexing, sorting and filling the material, and finally it is made available to the users in pigeonholes maintained in almirahs or in cupboards, which are decontaminated, and it is pest free and insect free.